So, myself, Dr. Sunil Avhad, the teaching concept topic of mine is Introduction to Cell Biology. Unit 1 is their cell biology, in which chapter 1, Introduction to Cell Biology, definition and scope, cell theory, generalized and prokaryotic and eukaryotic cell, size, shape, and structure is there. So, cell in Latin word, it is a small room. It's a functional and structural unit of all living organism. Cell was first discovered by scientist Robert Hooke in 1665. This is structurally how the cell is look like. It is carries various cell organelles, cytoplasm, plasma membrane. All these things are we discuss in upcoming slides. Modern cell theory, it is a very important when we discuss the introduction of cell biology there. So in modern cell theory, cell makeup of all living matter, all cell arise from other cells, chemical reaction of cell, anabolism, and catabolism take place inside the cell is there. Thus, cell is a fundamental unit of life. Types of cells, prokaryotic, lack of nucleus or membrane bound structure is there, example bacteria. Eukaryotic, have nucleus and membrane bound organelles, example fungi, plants, and animals are there. So firstly, we discuss about the prokaryotic cell having a lack of membrane bound nucleus, circular DNA, no histone is there, fluid structure has a cell membrane, as well as ribosome is there. Structurally or diagrammatically, we observe that the prokaryotic cell is there. Eukaryotic cell, it is a membrane bound nucleus, contain cell organelles, linear DNA, histone is there, unicellular to multicellular is there. So when we discuss the structure of the cell is there, this is the diagrammatically how the cell is look like. It is carries a cytoplasm. In cytoplasm, various cell organelles are present. Plasma membrane is there, cilia is there. One by one, we discuss in upcoming slides. So structure of cell, which carry plasma membrane, cytoplasm, nucleus, cellular organelles. So first one, plasma membrane, selectively permeable barrier to surround of the cell. By lipid is there, it is given by scientists Nelson and Danny. There it is described by fluid mosaic model, Singer and Nicholson. Made up of 3 micromolecules, mostly lipids, proteins, carbohydrates are there. This is the structurally or diagrammatically how the fluid mosaic model look like. It is made up of phospholipid, bilayer, glycolipids, glycoprotein, channel protein, as well as integral protein is there, extracellular fluid is there, cytosol part is there, as well as cholesterol is also in the diagram. This is the cytoplasm part. It is a fluid content of the cell, site for many metabolic pathway. Example, glycolysis, protein synthesis, fatty acid synthesis, purine synthesis. Nucleus is a control center. It is a prominent organelle. Most cells have single nucleus. Mature RBC have not observed the nucleus. Skeletal muscle cells have multiple nuclei. Nuclear envelope, it is a two membrane. Nuclear pore is there. It consists of circular arrangement of the protein surrounding the large central opening there. This is the control movement of protein and RNA across the envelope is there. This is the diagrammatically how the nucleus is look like. Function of nucleus is the contain DNA, direct cellular activities. It produces ribosome nucleus. Next, cell organelle is mitochondrion. It is elongated or rod-shaped. It is also called as the powers of the cell. It may be few as 100 or several as 1,000 depending on the cell activities there. This is the diagram of the mitochondria. So this is the function of this mitochondria. It generates ATP through the reaction of the electron transport chain. It helps in biochemical reactions such as oxidation of fatty acid. TCA cycle is there. This is the diagrammatically how the mitochondria is made up of. Ribosomes, it is a site of protein synthesis. High content of RNA is also. They are present free in cytoplasm, attached to rope endoplasmic reticulum. It consists of 2 units, large subunit as well as small subunit. Prokaryotes have 70S and eukaryotes have 80S. Diagrammatically, how the ribosome is made up of from large and small subunit is there. Endoplasmic reticulum. It is an interconnected network of tubules, vesicle, and system. It is extended from the nucleus to plasma membrane. 2 types of endoplasmic reticulum is there, rough and smooth endoplasmic reticulum. It is based on presence or absence of ribosome on their outer part. It is shown in the diagram also. Golgi apparatus, it consists of 3 to 20 cisternae. Small flattened membranous sac is there, is prominent in the cell that secrete the protein. It's also called as a sorting unit. It modifies salt package and transfer the protein from the rope endoplasmic reticulum. This is a Golgi apparatus structure is there. Lysosome, this is a membrane enclosed vesicle of the Golgi complex. It is a tiny organelle. It is also called as a suicidal bag. There is 60 kinds of powerful digestive and hydrolytic enzyme is there. It is rolling phagocytosis, rolling cell death, or autophagy is there. Various storage diseases like Niemann-Pick disease or Gaucher disease also. This is the diagrammatically how the lysosome is carry the hydrolytic enzyme is there. Membrane is there as well as transport protein is there. This is the last peroxisome cell organelle. Contains several oxidases. Peroxidase catalase is their function. Oxidation of amino acid, oxidation of long fatty acid chain, protect cell from the toxic effect of hydrogen peroxide. Mostly it is present in the plant seeds in the plant form. So thank you one and all for this presentation.